Assalamu alaikum everyone. Today we have an interesting topic to discuss, which is vertical gene transfer and horizontal gene transfer. Both of these topics explain very well the relationship between present organisms and their ancestors, that how the genetic information has been transferred between them. So first is the vertical gene transfer. In the vertical gene transfer, the genetic material is transferred from parents to the offsprings. By definition, transfer of genetic material from parental organism to progeny. Here we have example of cell division. In cell division, one cell divides into two daughter cells, and then these daughter cells give rise to another daughter cells. And this is the pattern that is following the vertical type, because upper level give rise to the next level and then next level. This is if the gene is inserted in the parent cell, that will also produce the product in the daughter cells. So that's why it is known as vertical gene transfer. Next is the horizontal gene transfer. In horizontal gene transfer, is of genetic material between unrelated individuals. Here we can see a gene has been transferred from one cell to another side by side. There is no need of cell division, so genetic information can directly be transferred from one cell to another. So it is following the horizontal type. That's why it is known as horizontal gene transfer. Next we have the examples of vertical gene transfer and horizontal gene transfer. In vertical gene transfer, we have reproduction as an example, because the cell division occurs in the reproduction. How? By asexual reproduction and by sexual reproduction. We know that in asexual reproduction, one cell divides into two cells by mitosis, but in sexual reproduction, two organisms have to come together to exchange their genetic information, and then new organism will be produced and the genetic information from parents to the offsprings will be transferred. And in the horizontal gene transfer, we have three examples: transformation, transduction, and bacterial conjugation. What occurs in transformation? In transformation, one cell give genetic information to another cell, and in this we can define transformation as the transfer of genetic material from one cell to another that can alter the genetic makeup of the recipient cell. So this is known as transformation. Next is transduction. You can see this is also an example of horizontal gene transfer, because the genetic information is being transferred from one cell to another. No cell division is occurring in them. So this transduction is only occurs by a viral vector. So this is side by side. That's why it is also known as horizontal gene transfer. Next, bacterial conjugation. In bacterial conjugation, there is a formation of sex pilus. From there, the genetic information travels from one cell to another cell side by side. No cell division is occurring at all. That's why this is also an example of horizontal gene transfer. But there is a difference between transformation and bacterial conjugation. What is the difference? You can see there is no sex pilus between them. There is no connection between them. Genetic information is transferred from one cell to another cell through the surface. But in the bacterial conjugation, a sex pilus, a connection is formed. This is the main characteristic feature of the bacterial conjugation that differentiates it from transformation. Next we have that how it is done when we are modifying the organisms. While modifying the organisms in vertical gene transfer, progeny produced from genetically modified organisms. In genetically modified organisms, we already know that one organism is modified by inserting the genetic makeup into the cell, and then it will produce an whole organism in next generation. This is an example that is given here is the Bt protein that is produced in the plants to fight the pests. And in the horizontal gene transfer, genetic information in genetic engineering is done to carry out the transfer of genetic material via a plasmid, in which one gene is inserted into the plasmid, and then a recombinant DNA, a recombinant genetic material will be formed that will produce our desired product in the next animal in which we will insert it. So there is no next generation, only the genetic information is being inserted, and this organism will produce our desired product. Next, via a viral vector. How it is done? Here you can see phages, bacteriophages, that is being inserted with the desired genes. Here you can see with the orange dots that are our desired genes. The products which will be produced by these genes is favorable, or we need this product. So bacteriophage will insert the genetic makeup into the target cell, and it will be attached with our DNA, desired DNA or targeted DNA, and then the product will be produced. How the antibiotic resistance activity is done by both of these processes. In the vertical gene transfer, it is gained by the offspring only if the parent possessed it, because if parents have an antibiotic resistant gene, then it will be transferred to the next generation. If parents don't have this protective or resistant gene, then it will not be transferred in the next generation. That's why it is necessary to have the resistant gene by the parents. While in the horizontal gene transfer, it is developed in the organism when an antibiotic resistant gene is successfully incorporated by it. When we inject a protective DNA in one cell, and it will transfer its genetic material to another cell. That's how the horizontal gene transfer will transfer our genetic makeup to the another cell, and that's how antibiotic resistant gene will be inserted into the other cells. So examples in eukaryotic cells. In eukaryotes, division of cells by mitosis and meiosis, simple example of vertical gene transfer, how the genetic material is being transferred from parents to offspring. It is the common example of VGT. So in horizontal gene transfer, now this is an interesting thing, that P aphids contain multiple genes that have been inherited from fungi, including those responsible for the synthesis of toluene. It has been studied that the aphids gets their red color. Here you can see red aphids, red color from the fungi. But how? They do not just eat the pigment and get this color, but they also inherit the toluene genetic sequences from the fungi. That's why they will produce the red color in their body and also in the next generations. Example in human. Transmission of HIV infection from mother to child is a common example of vertical gene transfer in the human. So if in the pregnancy, if the mother is infected with HIV, it is also possible that the child will have this HIV virus. But there are also 3 cases: in utero, during delivery, and breast milk. These 3 cases can cause HIV transmission from mother to children. And in the horizontal gene transfer, we have the example of papilloma virus, HPV, that produced worse on the body. Gene transfer of viral DNA by human papilloma virus causes cervical cancer in the human hosts. So the topic has been completed. If you have any question related to this topic, comment below. You can also download this PPT file from zulia3.blogspot.com. Link is also available in the video description. Like and subscribe.